So in today's session, we will actually continue with the immune response. Previous lessons, you actually look at the immune response. That means when a foreign antigen or a non-self antigen enter your body. So what will happen to your body? You learn about the primary immune response. Primary means when your body is exposed to the antigen for the first time. That is called the primary immune response. What does the cell of your immune system do? Okay, so then today we'll continue from there. So you learn that memory cells are produced, right, during the primary immune response. So we're going to look at the role of memory cells in the secondary immune response and long-term immunity. We are also going to describe the differences between active and passive, natural and artificial immunity. So these are the two topics we're going to cover today. The other two we'll cover in another session. Okay, so the first part we're going to cover is explain the role of memory cells in the secondary immune response and long-term immunity. So this is called immunological memory. That means they can remember after you have been infected by that particular pathogen for the first time. Subsequently, your body can remember. Okay, so how does it work? So this is a diagram where you use to learn about the primary immune response. Let's recap it first, okay? So number one is when you've got a pathogen that enters your body. So your body is being infected, all right? And this pathogen has got antigen on its surface. So therefore, we have got phagocytes will come in phagocytosderm. And then among the two types of phagocytes, neutrophil and macrophage. Macrophage will phagocytose and after that present the antigen of the pathogen on its surface. So number one, so this is actually step one in our immune response. Okay, after phagocytosis, there will be antigen presentation, followed by clonal selection of the B cells and T cells. Hang on. Okay. Clonal selection of the B cells and the T helper cells. Then after that, followed by clonal expansion of the selected T helper cell first. So during clonal expansion is when the cell will divide by mitosis and differentiate into active T helper cell and memory cells. Then active T helper cell will secrete cytokine so that it will stimulate the selected B cell to undergo clonal expansion. So during clonal expansion is when the selected B cell will divide and differentiate into the active cell is the plasma cell and the B memory cell. So we have got TH memory cells and B memory cells. Okay, and the plasma cells will then secrete antibody. And you have learned that antibody has got 6 ways of functioning. So one of the way is coating the antigen on the bacteria. So this is called opsonization. So this will promote phagocytosis. So this is the primary immune response that you have learned so far. So we are left with this part here where we want to continue to see what happened to our memory cells, why memory cells are being produced. So memory cells, B memory cells, T helper memory cells, T killer memory cells. So we'll start from here, okay? So this is a graph where the y-axis is the antibody concentration in the serum. So do you know what is serum? Can someone tell me what is serum? Daniel? So what is serum?
Serum. Can I give an example, teacher?
Can.
Serum is like anti-venom you get from venomous snakes, from the poison of a snake.
So it's an anti-venom that you use in the serum. So you are giving an example where you can use a serum which contains the anti-venom and introduce to the person. So what is serum? What is serum? Jing-er? Good try. Okay, good try. Jing-Er, what is serum?
I'm not so sure, teacher.
Okay, so for all of you, you already learned the blood, right? And the composition of blood. Tell me, blood is divided into two main parts. What are the two main parts? Jing-Er, start from Jing-Er first.
Plasma.
Blood is made up of blood plasma. Okay, plasma plus. Some more?
Vessels.
Blood vessels.
No. I don't remember, teacher.
Ayo. You must remember, when you study biology, this is called classification of things. You need to know how to classify if you want to do well in biology. Blood cells, isn't it? Plasma and blood cells. The liquid part and the cell part. Hello? You all got revise or not? Don't wait until exam time only revise. Revise when you have time. So plasma is the liquid part. Plasma can be further divided into a few things. What are the few things? Andrea?
Stuff like fibrinogen and those clotting things.
Fibrinogen and clotting things. So we are talking about the solute inside the plasma, right?
Yeah.
So give me example of solute in the plasma. Solutes.
Is it
Those are soluble in the liquid. That's why it's part of the plasma.
hormones? Was it?
Hormones is one of them, but you've got a lot of what are you transporting in your blood?
Like oxygen, carbon dioxide.
Yes, the useful substances, oxygen, carbon dioxide is the waste. So you're transporting useful substances as well as the waste. So apart from oxygen, what else is important that you are transporting in your
nutrients?
Yes, nutrients. Nutrients like you absorb from your digestive tract, like glucose, right? Amino acids, fatty acid, glycerols, all these nutrients, right? Cholesterol, etc. Correct? So nutrients. Then waste, CO2 is waste from aerobic respiration. What else we have? Waste like what?
Urea.
Yes, urea, nitrogenous waste, isn't it? Okay. So probably acid also. Never mind. Don't need to talk about acid. And of course, don't forget the important electrolytes are very important in our body. Sodium. That's why every day you have to take salt, isn't it? K+. These are three very important electrolytes in our body and needs to be transported in our bloodstream. They are very, very important for the functioning of our nerve cells and as well as kidney. Our kidney and nerve cell will need all this sodium chloride and potassium ions to function normally. So you don't take enough salt, then your nerve cells cannot function properly. Okay? So when you study, you don't wait. When you study, you have to at least within 24 hours revise. If not, as soon as possible, revise. And then try to write a summary chart and classify them so that you remember. Don't just read like storybook. As you study, you have to draw a summary chart already, a separate summary chart for yourself, so you can see things clearly. Okay? So that is solute, right? And of course, solute also will contain dissolved protein. So they are called the plasma protein. Right? Plasma protein, you've got a few classes. You've got albumin, you've got immunoglobulin, you've got fibrinogen. So 3 types. These are all in the PowerPoint last time you learned already. Okay? So this is the plasma and its composition. Of course, I forgot to say solute plus what? Solute plus water lah. Okay, solute plus the water. Okay? So then the blood cells you learn, you got white blood cells, red blood cells, as well as platelets are cell fragments. Cell fragments. Okay? So where does serum come from? If serum is in the blood, serum is which part of the blood? Chu Jiaying. Serum is which part of the blood?
The plasma, is it?
Yes, the plasma. But minus one thing from the plasma is called serum. What is it?
Minus plasma protein.
Plasma protein got so many types. Which type? It's only one of the different type plasma protein.
The fibrinogen.
Yes, fibrinogen. Fibrinogen. The reason is because, you know, some of you have taken your blood for a blood test, right? When you take the blood for the blood test, they'll put it in a test tube, and then it will be sent to the lab. And in the lab, analysis is not done immediately. So therefore, when it's placed, your blood is placed in the lab for some time, it will actually separate into two layers. The top is the clear liquid, which we call it blood plasma, and the bottom are actually all the blood cells. Right, red blood cells will be heavier, so it's at the bottom. White blood cells is the borderline between the serum and the phloem. Why heavier? Because a lot of hemoglobin with Fe2+. That's why it's heavier. Now this serum, this plasma is very interesting. If you continue to leave it there for some time, what happens is that you start to see, I'm not supposed to teach you today, right? You'll start to see a gel-like substance being formed there. So this gel-like substance is actually the fibrin. Fibrinogen exposed to the air becomes fibrin. So after fibrinogen converted to fibrin, what is left there, the liquid portion is called serum. So they actually will only take the serum for analysis. They will take the serum for analysis to check for your blood glucose level, sodium, potassium, chloride, and protein, and urea, and etc. So that is the serum part. Okay, so you need to know this. So you need to, when you study biology, you have to draw your own summary chart to classify the things and see it clearly. If you want to do well in exam, this is the secret. It's very easy. Okay. Okay, now we come back to this graph. This graph is graph of concentration of antibody in the serum. It's not nice. Concentration of antibody in the serum, okay, versus days. So what happened is that in day 0 is when antigen is being introduced to the person's body, right? Either by injection or you got infected naturally. So it enters the body, and the moment it enters the body, it will actually activate the primary immune response. It will start already. So primary immune response already start. So that means to say there's a lot of antigen presentation, clonal selection, clonal expansion, okay, until plasma cells produce and secrete antibodies. So from this time to this time here, you cannot detect antibody, but our immune system is very active. So that time where we cannot detect antibody is called the window period. Window period means the person is infected, but we cannot detect antibody yet. It's early stage of infection. So this is called window period. So even though you can make a blood test and say that this person is negative, it doesn't mean 100% negative, because you may be taking the blood during the window period where there's no antibody at all. Doesn't mean this person is not infected. So that is the window period. Okay. So after that, when plasma cells start to secrete antibodies, when antibodies start to circulate in the body, you will be able to find antibody in the blood, because circulation means it will circulate in the tissue fluid, in the lymph, and then in the blood. So you can find it in the blood. So therefore, the antibody level will start to increase. Okay, increase until maximum. So after increase until maximum, it will start to drop. So when it drops, you have to explain why does it start to drop. See, when you want to explain why a concentration of something in the body changes, you must always explain in terms of changes in the input and changes in the output. That overall causes the drop in the concentration. So input of antibody is produced by which cell?
Plasma cell.
Very good, plasma cell. Antibody is produced by plasma cell. The input is from plasma cell. Okay. Plasma cell. And the output, who will remove the antibody away? Sufyan.
Yeah, Teacher.
Who will remove the antibody away?
I don't know.
Okay, you look at this chart and tell me, who will remove the antibody away?
Is it the B cells?
No. Remove the antibody. B cells don't remove antibody lah. Look at the diagram, tell me who will remove antibody from our circulation that you cannot detect anymore. Open to the floor. Okay, open to the floor. Who will remove? Andrea Pang says neutrophil or macrophage. Very good. Okay, you see very clearly right in the diagram. First you must know what the antibody will do. Antibody must bind to antigen, right? And then there are 6 ways it will function, isn't it? You already learned already 6 ways it will function. So this diagram actually shows only one of the way, which is opsonization, right? It coats the antigen on the bacteria, and then it promotes this bacteria to be removed by the phagocytes, which is neutrophil and macrophages. So who will remove the antibody from our circulation? It is our phagocytes. So therefore, you need to write here, removed by phagocytes. So this is how you need to study biology, phagocytes. So this is called crystal clear understanding. So therefore, when you say that, okay, antibody level drops, right? Drop. So why drop? Very easy. Drop, it can be because no more plasma cell to produce antibody, because plasma cells are produced after some time, they will die. Okay, they have their own lifespan, so they will die. And then the antibody secreted, they are actually phagocytosed by phagocytes. Why? They already bind to the antigen. So they've already formed antigen-antibody complex, and then it promotes removal by phagocytes. So because of these 2 reasons, therefore antibody level drops. So you have got an empty graph in your notes. You need to fill in the blanks in this empty graph and pass up on next week, Monday. Okay, you have to fill up like what I have filled up here. Okay, you can take a photo of this so that you can refer to this when you do the homework. Okay, or you can fill in as I explain, but most importantly, listen to my explanation, you understand. You can always go back and copy. Take photo of this one and go back and write it down when you revise, okay? So antibody level will drop already, okay? So that's the primary immune response, right? Primary immune response, this will happen. Now, second time, you see here, subsequent antigen injection. Subsequent means after the first time. The second, third, fourth, fifth, etc., they are all considered as subsequent antigen injection after the first antigen injection or first infection. So when the same antigen is being injected again to the body, so the body immune system will be exposed to the same type of antigen for the second time. So in the second time, what happened is that you look at the graph, the antibody level increases. It starts to increase faster than the primary immune response. You see, it actually increased like this one, you know. Okay, and it actually increased, it takes a shorter time to increase. So the increase becomes faster. Faster than what? Faster than the primary immune response. And then the level of antibody, sorry, I cannot say the level, concentration of antibody is actually higher. You see, higher than the primary immune response. So there are 2 features here, or 2 properties here, is that the rate of the secretion of antibody is faster after antigen enters. It takes a shorter time for the antibody concentration to increase and increase to a higher level. Sorry, I cannot use the word level, concentration. Okay, higher concentration. So you need to explain why is it in this secondary immune response. Secondary means in the subsequent exposure to the antigen is called secondary, versus primary means first exposure. There is a faster synthesis of antibody as well as a higher concentration of antibody. So here, the effect is rapid antibody production and more antibody produced. Okay, I'll explain the rest later on. So why faster and higher? That has to do with memory cells already. Okay, that has to do with memory cells. So what does the memory cells do? What does the memory cells do? We use the previous slide to explain. I think it's easier here, okay? So here, let's say, for example, this is primary immune response where this one B cell is being activated to divide and differentiate into, let's say, this is too few already. Let's say you've got 5 plasma cells, right? So therefore, we assume it's the same thing. So we've got 5 memory cells, okay, equal number. So one B cell activated, divide, differentiate to form 5 plasma cells and 5 memory cells. So in the subsequent exposure to the same antigen, the normal immune response will also happen. That means to say that this immune response here will continue as usual. It will still happen. But now you got one additional thing, which is called the memory cells. Right? So because it's memory cells also will act. How does it act? Upon the subsequent exposure to the antigen, all right? So this memory cell will actually bind to the antigen. And after that, it will immediately divide and differentiate into more plasma cells as well as more memory cells. So what is the difference between the action of memory cells and the rest of the B and T cell is the memory cells don't have to wait for the cytokine from the active T helper cell to divide. The moment it's exposed to the antigen, it can divide and then differentiate immediately to plasma cells and memory cells, which means to say it will take a shorter time in order for us to detect antibody. Why? Because it can bypass all these steps involved before the cell can undergo clonal expansion. Upon exposure to antigen straight away, it can undergo clonal expansion. Okay, no need to wait for the T helper cell. That's why it's faster. So this explains why it is faster, because it bypasses all these steps here, right? So second thing is, why is it more antibody being produced, higher concentration of antibody? More antibody produced has to do with more cells, right? Right? Every cell, let's say every cell secrete maybe one mil. Okay, 1 mil is a lot, but never mind. Every cell secrete 0.1 mil. Then you've got 10 cells, needs to secrete 0.1 mil times 10, right? It has to do with number of cells that can secrete. So what happened here is, you see, in primary immune response, according to this diagram, one B cell activated can divide and differentiate to 5 plasma cells. So we've got 5 plasma cells to secrete the antibody. In the secondary immune response, other than a normal way of producing antibody, we have got additional cell, which is memory cell, which will also be activated to produce plasma cell. And imagine that one memory cells can produce 5 plasma cells, just like one B cell can produce 5 plasma cells. So one memory cell produces 5 plasma cells. Now we've got 5 memory cells to start off with. So 5 memory cells will give us 5 times 5 plasma cells. 5 times 5 plasma cells means altogether we have got 25 plasma cells that secrete antibody versus only 5 plasma cells in our primary immune response. Obviously, 25 plasma cells will secrete more antibody than 5 plasma cells, right? How many times more? 5 times more, right? 25 versus 5 is 5 times more. So you would expect the concentration of antibody secreted will be 5 times more, but actually it's more than 5 times. Because don't forget, the normal way of producing plasma cells are also being activated. This pathway also being activated. So altogether it's higher than 5 times. Okay, so that will explain in this graph, which you have to write into this graph. Okay, Why is it rapid antibody production? Because it can bypass number one. What is number one? T helper cells need to undergo clonal selection and clonal expansion, and then secrete cytokine. Only the B helper cell selected can undergo clonal expansion. So bypass number one is faster. So it's faster. Then why higher concentration of antibody produced? More antibody or higher concentration produced? Because we have got more memory cells to start with versus here, you've got less B cells to start with. So that's why these two reasons explain why in a primary immune response, the production of antibody is very slow. Slow because it's a delay to wait for T helper cell to undergo clonal selection and clonal expansion before our B cells can undergo clonal expansion. So that's why slow, right? Until when antibody is produced, it's very slow. Many steps involved. Then we got less antibody produced because we got less B cells to start with. All right, so this is homework that you have to fill in. Now, by the way, you don't have to know what is IgM and IgG because these are the classes of antibody not in your syllabus. You don't have to know. You just have to know that, okay, Ig stands for immunoglobulin. Immunoglobulin, which is actually equals to antibody. Finish. That's it. Okay, no need to know the detail. Okay, can So now let's do the quiz. Why memory cells persist for a long time? Say, for example, how many of you have got chickenpox before? When do you have a chickenpox? I think most of us got, right, in primary school. So once you've got chickenpox, it's said that for the rest of your lifetime, you will not have the symptoms of the chickenpox already. Why? Because your memory cells can remember once upon a time, your body is infected with that chickenpox antigen. So your memory cells remain in your body forever. Now you can ask me, so if you're 18 years old, right, you got your chickenpox in, let's say, primary one, right? So that means to say that now your chickenpox is it 10 years old already? No, your chickenpox is not 10 chickenpox. Your memory cells is not 10 years old. Okay, what happened is like this. The APC cell, antigen presenting cell that presents the antigen, they are long-lived, isn't it? So they continue to circulate in your body and continue to actually present the antigen on its cell surface membrane and the receptor. So your APC cell continue to move around the body, and then they happily present the okay, this is the remember, this is the antigen of the chickenpox. So it will present it, and as it presents it, your memory cells, you got receptor for it, will be able to be activated. So you know that the moment memory cells activated, it will divide, okay, and then it will differentiate into more plasma cell and memory cells. Okay, plasma cells and memory cells. Okay, so your memory cells are new. Your memory cells are new. It's not the same memory cells that produced at 10 years ago. They're being renewed. Okay, so therefore they can remain there for life. So that's why your memory cells are always there to be available for secondary immune response. Okay, now there's one thing that I skipped just now, which is very important. I'm bringing you back to this very important information, which is called symptom reservoir transmission. Please memorize this, symptom reservoir transmission. What does it mean? See, during the window period, right, when this person is already infected, but antibodies are not produced, it is during this period of time that this person will have symptom reservoir and transmission. Symptom means this person will get sick. Maybe you have got fever, you've got flu, you've got joint aches, muscle aches, okay, feeling weak and so on. These are the symptoms, or maybe diarrhea. Okay, symptoms. Why symptoms? Because at that time, antibodies are not produced yet. So the bacteria can be happily moving around your body, migrating everywhere, happily dividing to produce more cells, happily secreting secreting bacterial toxins, okay, and toxins enter your cell and then affect your cell functioning. So that's when you get sick. So there are symptoms. And at that time, because the bacteria or the viruses are still very active, you become, not you, the person becomes a reservoir. Reservoir means what? Wow, it's a good storeroom, you know. It's a storeroom that's moving around. So this person is carrying the bacteria that's alive or the viruses, going around to the restaurant, shopping mall, to the school, okay, and then start to spread to the rest of the people around. Why? Because they're reservoir. So this person is a reservoir because carrying all the pathogens alive. Okay, and because it's carrying alive and it moves around, it can transmit to other people. So transmission. So that means during the window period, this is when the person is not only sick, this person also is a reservoir carrying the active cell, the pathogen, and it can transmit to other people. So you have to avoid them as much as possible. Don't come to contact with them. Okay, because they can pass to you. So this is called symptom, reservoir, and transmission. Now, what about secondary immune response? What do you mean by that? In the secondary immune response, usually there will be no symptom, no reservoir, no transmission. No. Why no? Because in your secondary, in the secondary immune response, what happens is that when the antigen enters your body in the subsequent exposure, okay, subsequent exposure or infection, your body will launch secondary immune response when memory cells will quickly produce high concentration antibody. So antibody is very quickly able to attach to the antigen to carry out the six functions of antibody, like prevent entry, agglutination, prevent movement of the flagella, okay, causing lysis of it, causing opsonization and neutralization. So all these six processes. So it will actually prevent bacteria from migrating, from dividing, and the antitoxin from working. So this person will not be sick, and then the bacteria can die, either it's being removed by the antibody or removed by the phagocytes, okay, or neutralized by the antibody. And therefore, when all the pathogens are being removed, then this person is not reservoir and therefore cannot transmit to other people. So that's why we want, never mind. That's why we want to be vaccinated. Because when you are being vaccinated, that means we are trying to introduce the antigen of the pathogen, for example, COVID antigen introduced to your body in order to activate our immune response. For what purpose? For you to produce memory cells. So the main thing we want is memory cell, not the antibody, yeah? Because antibody level can drop, right? After some time, antibody disappear already. So what we want is memory cell, because memory cells will remain in your body for life, hopefully, so that next time you go shopping mall, next time you go back to Taylor's College, and then suddenly antigens start to spread happily to everybody. So when it enters your body, sorry, because you're already vaccinated, you got lots of memory cells, so you can produce antibody for very fast. You don't have symptom, you are not reservoir, you will not transmit to other people. There it goes. Goodbye, COVID. Goodbye, COVID, huh? Goodbye. Okay, so that's the power of vaccination. We want memory cell. Memory cell for what? Memory cell for long-term immunity. Long-term immunity. So all these are keywords that you want to write for exam. Memory cells, long-term immunity, secondary immune response. Okay, these are the keywords. Okay, now let's do the next question. Cepat. Okay, so we say, okay, so once I'm infected, right? Next time, when I'm infected, I cannot get sick. But how come I keep on getting flu? Okay, flu. Kindergarten got flu. Primary school got flu. Secondary school got flu. After that, go to uni, got flu. After that, start working, got flu. After that, become father, mother, got flu. Okay, why I always got flu, even though you're infected? Because, remember, we have done the question rhinovirus. Rhinovirus. right? Rhinovirus got many different type of strains, right? Many different type of strains. Many different strain means what? Each strain got different antigen. So that means to say that now it can be this virus that enter your body, this rhinovirus. Okay, and this rhinovirus, the antigen is triangular shape, say for example. Okay, triangular shape. So now all your memory cells produced can recognize this antigen. But too bad the next time when you're infected by rhinovirus again, there's a different strain. So probably it's a smaller virus, plus the antigen is not triangular, probably it's square, okay, or rectangular. Okay, so therefore, now again, this rhinovirus is this antigen entering your body for the first time. So you activate primary immune response, and during the window period, you can get sick, right? Because the symptom, reservoir transmission, memorize symptom, reservoir transmission. All these things are there during the window period. Okay. So this is called what? This is called antigenic variation. That means, okay, they are all rhinovirus, but then they've got different antigen because they're different strain. So that's why the person can get sick, because they can get sick during the window period. Okay. Now,